part today. We're going to continue with sex determination and have a look at how this works in Drosophila. So there'll be a nice compare and contrast to two different ways of doing it. And also we're going to come around and look at exon activation, which is a scenario that can occur in many different species that kind of addresses this problem that males only have one X and females have two of them, and a great number of genes on the X chromosome have nothing to do with sex determination, so you really want them to be equal in males versus females. So how is the organism going to grapple with that scenario? So in Drosophila, one similarity to mammals is that it also refers to chromosomes as the mechanism for sex determination. So it's entirely based on what genes are present and nothing to do with the environment. However, in Drosophila, they do have a Y chromosome, but that is pretty much irrelevant for the sexual phenotype. So that's different from mammals, which of course do rely on the Y chromosome. So in Drosophila, what we have here are 4 chromosomes. Number 1 is the X chromosome. So they may either have one X and one Y if it's a male, or 2 X's if it's a female. And then we have 3 autosomal sets. So 3 autosomes that come in pairs, a male, one inherited from dad and one from mom. So just like other diploid animals. So these are chromosomes number 3 and number 2. And then chromosome number 4 is actually really, really tiny. So in Drosophila, what's going to happen is that the cell assesses how many X chromosomes are present and how many autosomal sets are present. So normally the number of autosomal sets should be the same in every individual. That doesn't usually vary. You're going to have your set of chromosome 3, your set of chromosome 2, your set of chromosome 4. And then you ask how many X chromosomes, which is either going to be 1 or 2, depending on if you're male or female. So what the cells are doing is looking at the ratio between the number of X chromosomes and the number of autosomal sets. So in the female, they're going to have 2 X chromosomes. So here I've written 2 X's, and then 2 autosomal sets. So a set of number 3 and a set of number 4, set of number 3, set of number 2, and then this number 4 here is really tiny, so it doesn't much influence the ratio. So basically, we have a ratio of a 1-to-1 X's to autosomal sets. Okay, so that's the information that tells these cells to become female in phenotype. In the case of the males, the autosomal sets are the same, but we only have 1 X chromosome. So that actually is 1 X chromosome to 2 autosomal sets, or a 0.5 ratio, which is enough of a different kind of information to tell that to become a male phenotype. Now, just for contrast, if you had an XO autosomal male, so this would still become male because the male versus female phenotype is relying on the ratio, but it'll be sterile because there are genes on the Y chromosome that are ultimately needed for fertility, but not for sex determination per se. Okay, so here's an interesting little table for Drosophila, and one of the things about Drosophila is that you can really get in there and manipulate the genetics. So you can get these flies that have, freaky numbers of X chromosomes, like 3 X chromosomes, 4 X chromosomes, no X chromosomes, and or you can manipulate the number of autosomal sets. So you can tweak these ratios and get things that are less than 0.5 or more than 1.0 or something in the middle. And then you can kind of test our correlation here of the ratio of X's to autosomal sets being determinative for sex. And it holds up. So what we see is if we get a less than 0.5, we still develop as a male. It's called a metamale, and these are sterile. If we develop with more than a 1.0 ratio, these develop as a metafemale. They're also sterile. And then there are various different ways, as long as you have your ratio of having X's to autosomal autosomal sets come out to be 1, you're going to develop as a normal female. So it's not necessarily the absolute number of X's, but it really is referencing the ratio of number of X's to number of autosomal sets. So these all are 3 different scenarios where you still get female development if you have a ratio of 1. If you get something that's 0.66, which is right down the midline between the male ratio and the female ratio, that gives rise to this phenotype called intersex, which actually has some features that look more male in development and some features that look more female in development. And these are also sterile flies. So another difference in insects compared to mammals is that mammals are relying, especially for all the secondary sexual characteristics, on hormonal or endocrine-based mechanisms. So you have these signals that are flowing around the body, maybe made in the pituitary, going down and signaling the ovary, but they're more long-range signals that are communicating among the cells. In Drosophila, this is not the case. Instead, it relies on much more of a cell autonomous kind of mechanism. So each cell is acting independently, assessing what its genetics is, and responding to those genetics in an autonomous cell intrinsic sort of fashion. And so, again, this gives rise to the possibility of testing this by creating these animals that might have cells of 2 different genotypes. So these are known as gynandromorphs, and you can use some genetic tricks to create these animals that have some cells that are basically normal and have 2 X chromosomes, and then some cells that have lost 1 whole chromosome and only have 1, so they're really an XO situation. And then you can also use mutant genes to serve as markers for these chromosomes so that you can tell, which chromosomes are present in the cell. And in this case, we have an eye color phenotype and a wing morphology phenotype. As long as you have 1 wild type copy, that is your XX, you're going to develop as wild type eye color, wild type wing. But if you've lost one or the other of these, if you've lost the dominant allele, then you get this eosine eye and miniature wing, and you can tell the cells that have that phenotype because they are different with these visible structures. Or a similar situation in butterflies, they put some coloration genes on these chromosomes just to track which cells are which. So this comes out that any of the cells that have any of the regions of the body composed of cells of XX will develop with female structures, and then any regions of the body that have the XO phenotype will develop as male structures because they have a 0.5 ratio, and those cells think they're male, basically. So here's also some pictures of what the intersex phenotypes can look like. And these are referencing Drosophila, which you've had a chance to see in the lab. So if you can think back to the females, they had a somewhat larger, more pointed abdomen. And looking on the ventral side, up close at the tip, you can see the region where the eggs come out. This is called an ovipositor, and it's a kind of characteristic structure for eggs exiting the female body. In the case of males, their ventral structure, reproductive tract, is different. They have these structures called claspers that they use during copulation to kind of grab onto the female and hang on for the duration of copulation. And so these structures involve characteristic cuticle formations, and they're easily recognized. And then we have this intersex phenotype here that's due to a mutation in a gene, actually called intersex. And that gives something that's an intermediate between these two. So it's not quite male, it's not quite female, but something intermediate. Another one of the Drosophila sexual characteristics is the presence of a sex comb on the male foreleg. And this was something that was used during courtship, when part of the courtship is that the male flies go up and they tap female flies. And these stiff bristles are used to, I guess, communicate the presence of a male who's interested in courting. And so males typically have these, females do not have them on their forelegs. And then this 2 group 4 here, these are some mutants in the double sex gene where they have a reduced or increasingly smaller and smaller number of bristles developing as the sex cone. So that's also an intermediate phenotype for this organism. Okay, in mammals it was SRY that was a critical testes determining factor, and flies do not have an SRY gene, but they do have a critical gene, and this one is called sex lethal, abbreviated SXL. And here's a scenario. So when the X to autosomal ratio is 1, in this case, we're now going to make F sex lethal, and we will get, as a result of that, female development. Opposite scenario, if the X to autosomal ratio is 0.5, now no sex lethal is made, and we get a male development. And then here's some blockade experiment. So we have X to autosomal ratio is 1, but we have a mutation in the XSL sex lethal gene. So normally that should be turned on in females, and but if it cannot be turned on because it's a mutation, now we're going to get male development. Okay. So the only difference here is that in this wild type, sex lethal is made. In this mutant, sex lethal is not. And now we have female and male in the mutant situation. Okay. And then male is kind of control over here. Sex lethal mutation is still developed as males because they didn't need it anyway, so it didn't change anything for them. So we really believe that the sex lethal protein is something that's needed in Drosophila for female development. And so here's a little poem that's kind of a famous poem by Robert Frost. It talks about this path that's going in the woods, and we come to these points in our life where we might make what seemingly is a fairly small kind of random choice to go down one pathway and not the other, and then that's going to lead you to a whole different experience for the day and maybe for your life. And this kind of scenario can also happen during development, where what seems to be at first a fairly mild or innocuous choice to express this gene or not express this gene ends up having a long-term, fairly dramatic contrast. So there's a thought of a conception of something called a genetic switch, where you might switch the switch into the on position and make a gene product and get this whole phenotype based on that. Or the switch would be in the off position, you don't make the product, and you get a whole different scenario down the line. And sex lethal sort of fits that idea. So one thing about Drosophila is that since it's small and you can really grapple with the genetics, we know in great detail a lot about how the sex lethal gene is made and how it contributes to sexual differentiation. And one of the bottom lines is it's kind of complicated. So I'm going to try to just draw out some of the highlights here and sort of make the point of basically how this works. So sex lethal gene, when it's transcribed, of course, like all genes, is going to first be made into a pre-mRNA. So this is a long RNA that includes both introns and exons, and then we have to splice this RNA and make our final mRNA. And one of the things you'll see happening a lot in sex determination genes for Drosophila is they have alternative splicing. So we have here in the pre-mRNA 8 exons, including exon 3, which has a stop codon right in the middle of it. In the case of a female, this sex lethal pre-mRNA is going to be spliced such that that exon 3 is not included. So we go ahead and make a nice 1, 2, 4, 5, 6, 7, 8 mRNA, and then that can translate to make a functional sex lethal protein. In the case of male, due to the alternative splicing, the exon 3 gets included. There's a stop codon early in the gene, so even though there's an mRNA present, we never get any protein made, so no sex lethal protein made in males, which is what we want, right? We want sex lethal protein made in females and not in males. Okay, so when the sex lethal protein is made, now we have this differential situation where female cells have sex lethal protein it's functioning as a transcription factor, and so it's going to go to the next gene on down the line, which happens to be called transformer, and it's the same scenario. This pre-mRNA is getting made, and one of the early exons has a stop codon in it, and you get differential gene splicing so that the female version can make an intact protein from this intact reading frame. It goes all the way from exon 1, 3, and 4. So we get a functional transformer protein made. But in the males, even though they have the pre-mRNA, they splice it in such a way as it's impossible to make a protein out of the thing. And due to the presence of an early stop codon, now no transformer protein is being made. So that basically propagates this female-specific proteins being made and the lack of that in the males. Transformer 2 is itself a transcription factor, so it's going to act on double sex. And here, doesn't have to be an early stop codon, but we do actually get two different versions, two different alternative splicing products for double sex. So we can either get the female version of double sex through one mRNA, or the male version of double sex through a different mRNA splicing scenario. And then double sex has some jobs to do. And then there's also fruitless, which was another one of these genes that got mentioned in lab for being a gene that could be mutated and might affect courtship behavior as well as male morphology. So this is part of that sex determination pathway. So there's a whole hierarchy of genes, many of them are transcription factors. You'll see male-specific or female-specific splicing, and based on which proteins are present or not present, then that feeds into which targets get activated downstream or not. So here's just the whole figure put back together. This is in your book if you want to look back and read a little bit more detail of how this happens. Okay, next topic is dosage compensation. And this is basically a mechanism that animals have to deal with this fact that one of the sexes has 2 chromosomes, whereas the other sex only has 1. So females have 2 X chromosomes, males only have 1. And yet there's tons and tons of genes on this X chromosome that have nothing to do with sex determination, but still have other jobs to do in the body. So we need to make sure that the males have enough gene product, enough protein made from their 1X chromosome that they're able to survive and have normal metabolism. On the other hand, we don't want females to be overwhelmed with excess numbers of X-related gene products. So how can the body deal with this? And I'm going to show you 3 different animals, and they all do this in a slightly different fashion, but they still meet the goal in the end of equalizing gene products from the sex chromosome. So first, to Drosophila, the females, they have 2 X chromosomes that are transcribed. Genes on these chromosomes are transcribed at a particular rate. In the case of the males, since they only have one X chromosome, the rate of transcription from this X chromosome is greatly enhanced. So they actually get a double rate of transcription from genes on the X chromosome in the male genetic background. So that serves to give the increase in protein products so that these 2 can be equal. Does that make sense? It's kind of awkward to explain that for some reason today. Not enough coffee. Okay, we can do the same. We can meet that same goal in a different way. And C. elegans does this. So in C. elegans we have hermaphrodites, or males. And hermaphrodites actually have 2 X chromosomes, and the male only has 1. And what we see here is that in the hermaphrodites, relative to the other chromosomes, such as autosomes, transcription on the X is reduced. So they kind of shut this down and have a very wimpy transcription on the 2 X chromosomes, and that will balance out a normal rate of transcription from the X chromosome in the male organisms. So they get there by a different mechanism, but they meet the same goal. Okay, and then just to keep it interesting, mammals do it yet the third way. So they don't mess with the rate of transcription, they just pick one of the X's and shut it down. So that's X inactivation. And the females are the sex that have the two X's, so in females only, one of those X's will be chosen per cell. It will be shut down. It becomes this very dense structure known as a bar body that you can see if you stain the nuclei appropriately. And so these bar bodies are highly condensed and they're not transcriptionally active, and so then that leaves one X to be normally transcribed, which will equate to this situation in males where they also have one X that's normally transcribed. So this is X inactivation, and Mary Lyon was a scientist who was studying this and sort of figured out how it works. And what happens is that in the very young embryos, in the 1-cell stage, 2-cell stage, and for the first early cleavage embryos, we initially have both X's that are present and being actively transcribed. And then at some point in early embryogenesis, we have the event of X inactivation occurring, and one of those X's will shut down. And then for that cell, every daughter cell from that point on tends to shut down the same X. And so what this gives rise to is actually a patchwork of cloned regions. So in all these yellow regions here, one of the X's was shut down, and in these other regions it was the opposite X that happened to be shut down. So, and then those original founder cells propagated out, and all their daughter cells shut down the same X and kept the other one active. So in humans, you cannot really tell that we're patchy like this because the two X's don't have any different coloration associated with them, like skin color or coat color. But in certain organisms, you could tell. And cats are one of these. So if you happen to have a genotype of a cat where you get different coat color genes on the two X chromosomes, so if one X chromosome would have had the genetic information to make a black color, and the other chromosome would have had the information to make an orange color, then which X is still active will make a difference for the appearance of those cells in that clonal region. So you can get, this is how calico cats arise, because you have these different patches that are demonstrating which X is actively being transcribed in the organism. This is often combined with another gene called the white spotting gene, and that's influencing the migration of the neural crest cells. So a lot of cats have white paws and white ventral because the neural crest cells didn't migrate and you don't get melanocytes forming. But then over and above that, they have this X inactivation going on that will give you black patches or orange patches. So these different phenotypes. This also kind of makes sense of why calico cats are usually female, right? Because the male calico cats don't have X inactivation, so they're not going to get the patches. You can get a rare male calico cat if it's an XXY, because then that one still would have to inactivate one of the X's. Okay, so what are the mechanisms controlling X inactivation in mammals? And there is an interesting scenario that on the inactive X, really no genes are transcribed except for a very rare case. There's one gene called XIST, or exist, I call it, and this one is transcribed. In fact, it's transcribed only from the inactive X and not from the active X. So this is unusual. This exist RNA never gets translated into a protein, which is also unusual. Most of the time when we have an RNA that gets translated into proteins, mRNAs. And what happens here is that this exist RNA then seems to go to the inactive X chromosome that forms the bar body, and it kind of coats that chromosome, or it associates with the chromosome physically all along its length. So here's a picture of a chromosome that's the inactive X. Blue is showing you where the DNA is, and then there's a probe here in red for the exist RNA, and you can see how it's kind of coated or associated with the entire length of that inactive X chromosome. So this could perhaps set up some hypotheses to think that you need to have this exist RNA as part of the mechanism for either initiating or keeping the shutdown of this chromosome, inactivation of chromosome. So here's looking at some cells in a really early embryo versus slightly later. So as we said initially, both X's are on, and both of them are making exist and other RNAs. So initially, they're both making PGK, which is an X-linked gene, so you see two red spots. They both make exist, which is two light blue spots, and then this whole mass here in dark blue is the nucleus with all the chromosomes. So there's definitely two chromosomes in there that are expressing both of these gene products early. And then later, within the large DNA mass, one of them is only making exist, and the other one is only making the housekeeping gene, PGK. So they've sorted it out, they've made a decision, and one has become inactivated and the other is continuing to be actively transcribing. So what about these rare males, such as XXY? And here are a bunch of different genotypes from humans. Normal humans have 46 chromosomes and can be either XX or XY. And if you look at a probe for exist, so this is going to be a radioactive probe that can detect the presence of exist. In the case of XX, they're transcribing exist from the inactive chromosome, so it's present. Males are not transcribing any exist because they're not doing any X inactivation, so not present. And then here we have these males with multiple X chromosomes, and yes, there's plenty of evidence that they are transcribing exists, and they're going to shut down, in fact, all of the X's except for one, in the case for these multi-X genotypes. So that will bring them back to an XY situation. So before we really knew about the exist gene per se, we at least knew that there was an area in chromosomes where X inactivation starts. So initially these chromosomes are normal, they're all spread out and actively transcribing. But then as they shut down, you would lose gene expression starting from this one location and then spreading out in cysts to either side. And so this initial location was called the X inactivation center. It was the site where shutdown of gene expression started in the X chromosome that was going to be inactivated. And it turned out, lo and behold, that is the site where the exist gene maps. So that's the location on the X chromosome where the exist gene is located. If you take that exist gene and you put it on an autosome, you can get the autosome to undergo X inactivation, or inactivation, if you will. So you can get the shutdown of genes located contiguously to that exist gene to now no longer be transcribed. So there seems to be something very special about the exist gene and the fact that it's going to act on chromosome that is in cis, or literally next to it, contiguous with it, and leaves the rest of the chromosomes out there in the genome not affected. So how does this work? It seems to involve what we broadly term epigenetics, or additional kinds of modifications that occur in the chromatin. So chromatin, as you know, is composed of DNA that's coiled around histones, and there are a lot of different covalent modifications that can be done both to the DNA or to histones that affect how open this chromatin structure is. So that means how accessible is that chromosome to the polymerases that could go in there and start transcribing products. And two key types of modifications are acetylation, where you take an acetyl group, you stick it on there, or methylation, which you take a methyl group, stick it on there. So they're not that different chemically, but they are just different tags that you can put either on the DNA or on the histones. So in the case of exist RNA, an inactive chromosome, we see that it's methylated, these purple dots, and deacetylated, whereas the more active form of chromosome that's associated with RNA pol 2 that's doing the transcribing that one's going to be acetylated but not methylated. And you can also do stains that will pick up these signals in the chromatin. So only one of these guys is showing you the phenotype associated with the inactive X. Okay, so this is to make sure I told you about the mechanism of sex determination that is not reliant on sexual chromosomes and instead relying on temperature. So here we have a few different organisms, including the turtle, the alligator, and this guy. I'm not quite sure exactly what it is. And the sex ratio is located on one axis and temperature on the other. And so for the case of something like alligators, there's only this narrow window of temperature from 33 about that is going to give you a high ratio of male development. So if you're too high or too low, that signals that egg to develop as a female. So you have to be exactly 33 degrees if you're going to be a fertilized egg and develop as a male alligator. And then other organisms also rely on temperature, but they have their own readout of the environment, their own readout of specifically which temperature means what. Okay. So one of the things is that if you start out with an environmental cue for sex determination, at some point that still has to translate into how are we going to get differential gene transcription, right? And then we have to bring it back to the males need some genes and females need a different set of genes because we're making different morphology here. So how can that work? And one of the main take-homes is that it does seem to play into the endocrine system. So the temperature is affecting hormones that are made within the body, and you can get different hormones if you're male versus female. So a male hormone is testosterone, a female is estrogen. Biochemically, these structures are not that different, and you can have this enzyme known as aromatase that can actually convert the testosterone into estrogen. And so how much aromatase activity is present is going to make a difference for male development versus female development, because it's taking your starter building